So this is going to be the third lecture I have for you on biotechnology. And in it, we're going to talk a little bit about genomic libraries, this thing called DNA gel electrophoresis, and we'll talk a little bit about how DNA fingerprinting works as well. So I wanted to first talk to you about just this general thing called the genomic library. And a genomic library is a way that we can actually store DNA to analyze later. So what we'll do is we'll create what's known as a plasmid library, where we take DNA, say we'll take the entire DNA from an organism. We want to be able to study it later, but we just want to hang on to it. And so what we'll do is we'll cut the DNA up using restriction enzymes. We'll insert the DNA into plasmids, and then we'll give those plasmids to bacteria. So now all of those bacteria together contain the entire sequence of DNA from an organism. And we can then use that genomic library later to study the DNA. And this is, we'll talk about this, this is how the human genome was actually sequenced. So it's a way of storing DNA in a DNA library. Now another technique that I'm actually going to have you guys do, virtually anyway, is this process called gel electrophoresis. And this is a way that we can actually separate DNA so that we can analyze the DNA. This process works because DNA has an electrical charge. DNA has a net negative electrical charge. So if you were to put it in an electrical field, think about the electricity generated from a battery, where you have a positive end of the battery and a negative end of the battery. That tells you the ends or the charge of that electrical field. If you were to put the DNA into that electrical field, it's negative, so it's going to move towards the positive end. So what we do is this. We create this device. This is the gel electrophoresis device. It's basically a gel material. Now, the gel material is made of lots of different kinds of materials, but we usually use either a starch material or a plastic material. And when you produce these materials, we'll use starch. The starch forms a mesh. It kind of looks microscopically like a sponge, but I want you to think of it more like a colander. So if you ever make spaghetti at home, I don't know if any of you break your spaghetti, but if you break your spaghetti, so you get some larger pieces and some smaller pieces, when you go and dump the spaghetti into the colander, the smaller pieces tend to work their way through the holes a bit faster than the bigger ones. And so if you were to continue to shake that colander, so really give it a good shake, the spaghetti, little pieces start coming through the holes, and they'll go through the holes much faster. So we use that same process here. This gel material basically has microscopic holes. So we build this gel, it's actually heated, so if you've ever made Jell-O, you heat it up like you make Jell-O, and then it gels, it congeals into something solid. And so what you do is you leave a device in it called a comb that leaves indentations. So you pull that comb out, and you're left with indentations. And then you put the DNA in those indentations so it actually runs through the gel. And typically what we have to do, if you look at this image here, you have to stain the DNA because the DNA is clear. So we use a stain that actually glows. It sticks to the DNA and it glows under UV light. So you can see these orange stripes. Those are the different pieces of DNA. Now, here's where this comes in, and this is why it's useful. As the DNA runs through this, so I've got an electrical source attached to it. This is the negative end. This is the positive end. If I apply electricity to this whole thing, and by the way, this whole thing sits in basically a salt water bath to conduct the electricity, the DNA gets pushed through the gel from the negative towards the positive. The really small pieces move quickly, so they're going to go migrate through the gel faster than the bigger pieces. The bigger pieces are going to move more slowly, and they'll be at this end of the gel. Now, this is actually what it looks like. These are some images that I have from a job I used to work at. And so you can see here, up here, these black rectangles, those are the spots from the well. And you can see right here, these are different fragment sizes of DNA. So we ran DNA in the gel here. This piece is very big. The pieces down here are very small. And so it's broken up by fragments. Now here, if you look at these things, these are DNA molecules that have been cut up with restriction enzymes. And so it looks like a smear because we get all different kinds of pieces, lots of different sizes, from very big to very small. And since we have lots and lots of different size pieces, we get a smear. Now down here, you can see all the DNA. You have these really bright bands. Those DNA fragments are all about the same size. This is actually a genetic test. We were looking to see, these are fragments, by the way, created by PCR. The fragments are all the same size, so they don't have any differences between them. Here's how we can use this. We can use gel electrophoresis and PCR to create a DNA fingerprint. Now, one of the ways we can do this is we use these things called restriction fragment length polymorphisms, or RIFLIPs. These are basically different DNA sequences where we'd have different restriction enzyme cutting sites in the DNA. And if you have a mutation, so this individual has a mutation, they don't have that restriction enzyme site. And so when you go to cut up their DNA, or when you amplify it with PCR, you get different sized fragments out of their DNA. So if I run this individual on a gel, they have two fragments. They have a big one and a small one. This individual only has one big fragment because they have a mutation here. And so when you run them on the gel, you get different banding patterns. We can use those different banding patterns to create a DNA fingerprint. So that's what this is. This is actually showing you these are DNA fingerprints. Now the black lines represent different sized bands of a gel. We just produce the image a little differently, so the lines are black. But you can actually use them to actually compare. So if you're comparing a mom here and her child, you can actually try to figure out who's the father by matching the DNA fingerprint from these males to try to identify that. Now, here's an actual DNA fingerprint. This is what one looks like. My DNA is actually on this gel. I just don't know which lane it is. But it's showing you in DNA fingerprinting, we basically get a group of bands. So these bands all represent different sized fragments in the DNA, and they can indicate different kinds of mutations. And you can see that everyone sort of has over here, they all have this band, but if you compare these two individuals here, you can see there's a slightly different banding pattern. So right here and right here, those fragments are not the same size. So that indicates a genetic difference between these two individuals. So I could take DNA from an unknown individual, compare that to a known individual, and if the banding patterns matched, they would most likely be the same individual. Now, the last kind of technique that I just wanted to mention is this thing called a DNA microarray. And this is used to actually look at gene expression in a cell. So what we can do is we can actually screen for thousands of genes at once. So we can look at to see which genes are being expressed in a cell. And this is really useful if you're trying to figure out how a drug affects gene expression. So what we do is we will isolate the RNA out of a sample. We'll create cDNA out of that RNA, and we'll label it with a fluorescent marker. And then we'll apply that to a glass slide. And on a glass slide, we have these blue dots. Those blue dots represent what's known as probe fragments. So we have these pieces of DNA that each dot has a very specific sequence. So if the cDNA, if the sequence matches the dot, the sequence of DNA on this dot, they'll stick together. And when you shine a light on that, those regions light up. And so this is showing you a DNA microarray. So what we can do is compare two DNA samples from two different tissues and see if I add a drug, which genes are turning on and which genes are turning off.